Complete human tissue regeneration has been a driving force in modern medicine. Burn, amputation, and severe injury treatment would all benefit a great deal from the ability to control human tissue regeneration. There are plenty of examples in the animal kingdom of animals regenerating entire tissues completely. So, why is human tissue regeneration so limited? Many of the examples in the animal kingdom directly defend these animals against predators. For example, salamanders have been shown to lose and regrow entire arms. Mice have some limited capacity to lose and regrow their tails in response to predation. So how do they do it? Some scientists have suggested that blastemal cells present in many of these animals play a critical role. Other scientists have investigated the role of cell cycle control elements such as the gene P21 present in specialized cells in many of these animals. Perhaps investigating other examples of tissue regeneration in nature will yield more clues as to its origin and potential for control. While others in her field were investigating other animals, Ashley Seifert and her team had heard rumors about a small African mammal known as the African spiny mouse. These unusual animals were rumored to exhibit strange properties when caught, including tearing of their own skin. Ashley Seifert and her team want to know just how far these abilities went and how they worked. We wanted to know if those abilities could be used one day in humans. To that end, we've gone to the University of California, Irvine to talk to some proxies for Ashley Seifert and her team.
We observed that the African spiny mice exhibit skin autonomy, followed by a fascinating regeneration process. First, we created a full-thickness excisional wound, which is a wound down to the subcutaneous layer, of an area of about 1.5 centimeters squared. After 30 days, the new tissue is indiscernible from the native tissue. More interestingly, we saw that this tissue regeneration occurred even at the hair level of the hair follicle, an extremely rare and sought-after feature.
So we compared the spiny mouse to a regular mouse, which we used as a control mouse. It's important to first mention that the spiny mouse can only handle a small stress on its skin versus the normal mouse. This tells us that they likely have much different structures that comprise their dermis. Here we use stains to compare the mice skins. We saw that the spiny mice have much larger hair follicles and that keratin-producing cells do not migrate into its new wound sites. Nor does it have densely packed collagen structures running parallel to the wound. Its collagen is loosely packed and more disorganized. Already we're starting to see what makes its skin weak. After looking into embryonic signaling pathways, we decided to compare the presence of signaling pathways known to be involved in hair follicle development. The first was the WNT signaling pathway, which is a requirement for normal follicle formation. It was observed by detection of the protein LEF1, which localizes WNT signaling. It showed that there was a high localization of the protein with the regenerating hair follicles and matrix cells of the spiny mice. This was not seen to the same degree in the control mice. This leads us to believe that it may partially account for the follicle hair regeneration.
Another signaling event taken into account was the BMP signaling pathway, which is known to be activated during embryonic development. This time, proteins known as SMAD1, 5, and 8 were used to localize the signaling pathway BMP. We observed similar results to the WNT signaling pathway in that the high levels of SMAD proteins were detected in the spiny mice tissue samples relative to the control groups. This means that the regeneration of hair follicles in spiny mice has refined steps, exhibits high proliferation, and redeploys embryonic molecular pathways, which was astounding to say the least.
We created ear punch holes in both of the mouse species, and we saw stark differences in their ability to regenerate completely this forlorn tissue. We saw this at all levels down to the cellular. Spiny mouse showed complete regeneration of the wound site, including hair follicles, cartilage, adipose tissue, whereas the control mice did not. At the cellular level, we discern why this may be. Cellular proliferation exists continuously at the proximal wound site in the spiny mouse until the tissue regenerates. When looking at the myofibroblasts, which specifically designate a low ability to regenerate, we see none at day 12 in the spiny mice versus the control mice. Overall, at day 30, we see that the spiny mice have regenerated their tissue, and it looks similar to the native tissue. However, for the Control mice, all we see is a missing spot and some regeneration.
Our conclusions so far led us to look at differences in the extracellular matrix formation of the spiny mice compared to the control mice during regeneration, and what we found was very discerning. Essentially, what we found was that the regenerating tissue of the spiny mice kept an important balance between dermis reformation, or scarring, and maintenance of a pro-regenerative wound epidermis, whereas the control rabbit ear, in lieu of formation of a wound epidermis, formed a stable basement membrane and epidermal scarring. This makes sense because the epidermis of the spiny mice was so easily detachable in the first place. Any future work done on this topic would most likely stem from an investigation into how this fibrosis is exactly controlled in spiny mice.
Wow, such a fascinating study. With skin and hair regeneration of African spiny mouse, we begin to ponder if it is a possibility for us to make use of regenerative abilities in the future.
The wars in Iraq and Afghanistan have resulted in more than 1,500 U.S. military amputees, many more with lost eyesight or severe burns. DARPA, an agency of the U.S. Department of Defense, has set up AFIRM, Advanced Forces Institute of Regenerative Medicine, to study the possibilities of regeneration, focusing on burns and limb salvage treatments in humans. With the increasing knowledge of regeneration from animals, AFIRM has created a more efficient method of treating burn victims without the use of skin grafts. Instead, a spray gun containing the patient's own stem cells are sprayed over the burn area, followed by an extracellular matrix membrane placed over the stem cells. This method results in power healing with almost full recovery, but not to the extent of the African spiny mouse. Perhaps what we learned in the African spiny mouse can be applied to humans one day in regenerating hair and skin.
Poof, there go the car. Poof, there go the crib. Poof, 100 mil. David Copperfield. David Copperfield. David Copperfield. Poof, there go the car. Poof, there go the